In the previous video, we demonstrated how to use the RAST website, how to set up an account and submit genomes to RAST using both the classic RAST pipeline and the RAST toolkit. In that video, I showed you how to download a config file from NCBI, which will be a prerequisite for this video. So in this video, I'm going to demonstrate how to run the RAST toolkit scripts from the command line. So the first place I want you to navigate to is a web page called tutorial.theseed.org, and that's all lowercase, and the seed is one word. This will bring you to an important web page that we use for keeping written tutorials, workshop agendas, and links to other important resources. The first link that I want to draw your attention to is the RAST Toolkit DMG. It says download the RAST Toolkit DMG, and this is how, if you're a Mac user, you can download and install the RAST software on your computer. And if you click on that link, it'll bring you to our GitHub page, and you can download the DMG by clicking here. When you click on this link, it'll take you through the steps of installing the RASTtk scripts onto your computer, and the application will bring up a command line window with the correct environment for running all of our scripts. For the current demonstration, I'm going to demonstrate all of the scripts in the Iris environment. So if you go back, the link above here is for Iris. And Iris stands for Interactive Remote Invocation Service. So go ahead and click on it. And it is a JavaScript application that runs in a browser window. And it has the behavior of the command line. It's like a pseudo command line, except that there's no installation of software, and it's really ideal for demonstrating software to large groups of people. So you should navigate to Iris and follow along this tutorial using the Iris interface. But if you have decided to download the RAST DMG, if you bring up the command line window, all of the commands will be identical and the behavior will be identical. So you could also follow it there as well. So the Iris environment looks like this, and in the main panel is an area called the command console, which will give you output from jobs that you run and status and so forth. On the left side contains an entire list of scripts that we commonly use, and the scripts that we're going to be interested in are under Annotation. And if you scroll down, you can see all of the annotation scripts that are available that can be run in the Iris environment. And the RASTtk scripts all begin with the prefix RAST-. When you run a command, down below in the running processes window, you'll see that command running after it's been launched. And there's a file browser where you can see the files that you've created and click on them and download them. So the first thing I'm going to have you do is sign in, and this will take your RAST credentials. So click the sign in button in the top right corner, and I'm going to sign in. Oops. Okay, and then it'll say if you've signed in correctly, authenticated, and your name. So down here is the command window for Iris, and it works like a Unix command line. So you can run commands in it like ls, and currently there's nothing in our workspace except a file called history. So the first thing that we're going to do is we're going to upload that contigs file that we had downloaded in the previous video. So I'm going to navigate and grab that file and bring it up. So the upload file is this up arrow in the right hand corner, and when that shows up, you'll be able to see that file, the contigs file, in your file browser. The next thing that I'm going to do, since this is actually a workspace system, you can make directories to work in. So I would like to make a directory in order to work on our particular annotation job. So I'm going to type the command mkdir. That's the Unix command for make directory. And I'm going to call this RAST TK demo and hit return. When you hit return, over here in the file browser, you'll see a folder called RAST TK demo. And now the next thing that I want to do is I want to move our contigs file into the RAST TK demo. To do this, we will type the Unix command mv, which means move, and our file name. And you can cheat by moving the file name, propagating the file name into the browser, into the command line, by hitting this right arrow key, or this right arrow button. So mv, our config file name, and then RAST TK demo, and hit return. Then you'll get this message in the command console that says that it moved NC00913 to the RAST TK demo folder. And over here, if you click on the folder and open it, you can see that we've moved our config into there. Now, we're currently working in the directory above this, so we actually want to navigate to the RAST TK demo directory because that's where we want to work. So we will say cd, which means change directory, and then we'll type in RAST TK demo. And then it says change directory to RAST TK demo. So if you do an ls, which means list, you'll be able to see that the only file you can see now is our config file. So when you're working in Iris, there are shortcuts that you can do in order to see the programs that are available to you. And I just mentioned that all of the RAST TK scripts begin with the prefix RAST. So if you type RAST dash and then hit the right arrow key on your computer, on your keyboard, this will list out all of the available RAST TK scripts in the command console. So I'm going to demonstrate the default RAST TK pipeline in this video. And the default RAST TK pipeline uses 3 scripts, which I will demonstrate each of them, and then you'll be able to download your formatted the file in the format that you wish to have it in. So the first RAST TK script that you need to familiarize yourself with is called RAST create genome. So let me see if I can find it. Here it is. So if you click on RAST create genome, it will propagate that name down into the command console. Now each of the scripts for the RAST toolkit contains a help menu option, which is you can bring up by typing the script name and then -h. So I'm going to bring up the help for RAST create genome. Okay, here's the help menu for RAST create genome. Now, in order to understand what's happening here, I should regress and say that the command line scripts for the RAST toolkit behave in the same way that the website does. So you have to, when you submit a genome, you have to submit the metadata associated with it so that RAST knows how to call the genes and so forth. So when you submit your genome, you have to submit it with information including the genetic code, the domain, the scientific name, and obviously what the contigs file is. In this case, we submit those data in a special file format that's a JavaScript file format called a genome typed object. So this first command, RAST create genome, you're actually building this genome typed object on your computer. So I'll show you how to do that. So in the command window, we'll type RAST-create-genome. Now the help menu for this shows you all of the different program options. And when you declare a program option, it has to be declared verbatim as it's written. So the first one we're going to use is scientific name. So I'm going to type RAST create genome -- scientific, all lowercase, -name. And this was an E. coli K12 genome. And we have to use quotes in this case because there are spaces in the E. coli K12 name. And if we don't put it in quotes, the program won't parse it correctly. So Escherichia coli K12, end quote. So there's the first option. We've just given RAST create genome the scientific name of the genome that we are about to annotate. The next option is the domain. So -- domain, all lowercase. And in this case, it's bacteria, capital B, bacteria. Now we don't need quotes here because this is all one word, whereas here we had multiple words. The third option is the genetic code. So it's -- genetic code, genetic - code. And the genetic code value for E. coli K12 is 11. Remember, these are the NCBI taxonomy genetic code values. Finally, we have to submit the contigs file that goes along with it. So -- contigs. And that's this file over here. So I'll actually cheat and hit the right arrow key, but we have to delete the directory name that was brought along with it. And so now we have RAST create genome, scientific name is E. coli K12, the domain is bacteria, the genetic code is 11, and the contigs are NC00913. Now, we have to tell it to make a file from that, and so the greater than sign means send the output to standard output, and we will create a file name for this, and I will call this E. coli.gto because this is building our genome typed object file. And then I'm going to hit return. Now, as it's running, and it ran almost instantaneously, there was a little spinning wheel here, and the process showed up in the running processes window. But it's run already, and now we have a new file over here in the file browser called E. coli.gto. So I'm going to go ahead and click on that. This brings up the GTO file in a new window, and I just want to show you what it looks like. It's JavaScript formatted, but it basically just contains the information that we just entered. So here's the scientific name, here are our contigs. And if you scroll to the bottom, you can see the ID, the domain, the genetic code, etc. So I'm going to close that. So now we've created the file that the RAST TK web server is capable of ingesting. So RAST create genome built our genome typed object. Now it's time to move on to the next script. So I'm going to do RAST dash and hit the right arrow key again so that we can see them. The next script is called RAST process genome. So I'm going to click on RAST process genome, and that's going to bring that up down here in the command window. And again, I'm going to show you the help option for that using -H. Okay. RAST process genome actually doesn't have very many dependencies, so we actually can run this one straight away. So I'm going to type RAST dash process dash genome, and I'm going to use the less than sign, which means read from standard input. So here's how I know to do that. And our input file isn't the contigs, it's the genome typed object that we just built. So E. coli.gto. And I will make an output file now called E. coli.anno.gto. And I'm just naming it this way so that I know what I've created. They're both genome typed object formatted, but this new one will contain the annotation information that we that the RAST toolkit finds. So then that's all that this requires, so I'm going to hit return. This launches our RASTtk job. So you can see that we have the spinning wheel saying that the job is running. We can see that we're running RAST process genome over here in the running processes window. And this could take as long as 5 minutes, so this might be a good point to pause the video if you're working with a large group. There's one more thing that I would add, and that is that this does not submit a RAST job into the typical RAST queue. It uses a different computational infrastructure, so there are times when it's very typical that one of these jobs will run much faster than a job where you have to wait in the queue. So I have a tendency to annotate on here pretty often. Okay, it's been about 5 minutes, and you can see now that there are no processes running in the running processes window, and we get this nice message saying that the command completed in the command console. And if you look under here, under our file browser, you can see in the RASTtk demo folder, we now have a file called ecolianno.gto. And I'm going to click on that so we can look at it. Okay, now initially this looks just like the genome typed object that we had before, except that now it's a bigger file, and if you scroll down, you can see that the annotation information has been stuffed into this file. So as annotation programs ran, they added their data to this file. And so you can see the protein translation, for instance, the location information, the functions, and you can also see which gene caller called them, and so forth. So I'm going to close out of that, but this shows that our annotation job did in fact run. So we've just run 2 commands. The first was RAST create genome, which created our genome typed object, and RAST process genome, which sent our genome typed object to the RAST server and where it got annotated. And now we have our annotated genome typed object in our file browser, but it's not in the format that we want. So we need another RAST command. And so I'm going to do my little trick where I type RAST dash and hit the right arrow key. Now we need the command RAST export genome. So I'm going to click on that and show you the help option for this one. RAST export genome allows you to export your genome in a variety of formats, including GenBank, GenBank merged, FASTA, etc. And I'll step you through a couple of use cases for this program. So I want you to type RAST export genome, and we're going to first ask it for the protein FASTA. So I'm going to say protein FASTA, and now I will read our annotated GTO from standard input, so E. coli.anno.gto. And first, I'm not going to actually create an output file. I'm just going to hit return. This will run fairly quickly. And what this will do is it will show you the output in the command console. And you can see now that what we asked for down here, if we hit the up arrow key, we can see the command that we just wrote. We asked it to give us protein FASTAs, the protein FASTA for the E. coli annotation. And you can see that that's what we got. And our version of a FASTA contains the annotation as well, and in some cases the EC number. Unlike the command line, you'll notice down here that the output gets truncated, and that's because the JavaScript will chew up a lot of memory on your computer if we gave you the whole genome. So what we want to do is we want to create a file for this protein FASTA, and so we will call this, we'll do the greater than sign E. coli dot proteins.fasta. Now when we hit return this time, it will create the complete file over here in the file browser. And you could click on that if you wanted to. The other button that there are two other buttons that I would like to point out. The first over here is the pencil, which allows you to edit it. So if I clicked on that, it would bring up the protein FASTA over here in this other tab, although it's taking a while to load, so I'm going to go ahead and close it. But if you wanted to actually edit the annotation before you downloaded it, this is where that would happen. The other button I want to show you is the download button, and that will bring up a downloading menu in which you can determine how you want this to end up on your computer, in which case you would be done if this is what you wanted. But I will cancel out of that. So let's say you wanted to get the protein encoding genes in FASTA format. Let me show you how to do that. So we'll go back to RAST export genome and look at the help. In this case, you'll notice that there is no supported format for protein encoding genes in FASTA format. So what we have to do is we actually have to declare the feature type. In the RAST world, we call the things that were called in the config the features. And the feature type that we want in this case are the CDSs, which are the coding sequences. So the syntax for this would be RAST-export-genome -feature-type, and it's CDS, all uppercase. And then we have to say feature DNA right here, because we're asking for the DNA associated with the CDSs. Our input is our original annotated genome typed object, so E. coli.anno.gto. And our output we will call E. coli.genes. Well, we better call it what it is, cds.fasta, and hit return. Okay, that brought up our CDS file over here in the file history, and I can say, I can use another Unix command such as head, and then E. coli.cds.fasta, and hit return, and it'll show us the first 10 lines of this file to show that it in fact did run. And so here are the genes, here are their annotations, and their EC numbers and so forth. So we got a DNA FASTA file there for the protein encoding genes. Now, in order to run that command, we needed to know what the feature types were, and I'm going to show you how to do that. So if we go back to RAST export genome and go to the help menu again, you can run the command. I hit the up arrow key to get back to the previous command. And if you ask for the feature data, this will give you the table of all the features that were called in the genome. So I'll type RAST export genome feature data, and I'll read our original annotated genome typed object from input, E. coli.anno.gto, and I'll hit return. This shows us the truncated table of all of the features that were called in the genome. Now, in order to see what different, so the feature table is in the format, here it has a gene ID, the contig on which the gene was found, and its location, the feature type, which is CDS here in the third column, its annotation, and this long string is called an MD5 value, and we use those to determine things that are identical. So you wouldn't use that unless you're into a more advanced use case. So I'm going to show you how to get the different feature types. So if we do that exact same command, we'll hit the up arrow key to get back to that. I'm going to create a pipe. That's the vertical bar character. And I'm going to pipe this output into a cut. And cut is a Unix command that will take an individual column. And I'm asking for the third column, so it's actually the third field, so it's -F3. And I'm going to pipe that output into another Unix command called sort. And the -U option on sort sorts unique. So now what I've asked for is every member of this third column. I've asked for this table, every member of this third column sorted uniquely so that I can see the different types that are there. And then we'll hit return. This shows you the different feature types that the RASTtk default pipeline found within our genome. So we found CDSs, which are protein encoding genes. In previous implementations of RAST, we've called those PEGs for protein encoding gene. The CRISPR array, the CRISPR repeat, the CRISPR spacer, repeat regions, and RNA genes. So now if we wanted to, instead of the previous, so we can page back through the history by hitting the up arrow key. And if we want, now I'm back at this original command where we were asking for the CDSs, and I can fix that. I'll delete the output, and then I'll come over here, and instead of CDS, I'll say RNA, all lowercase, and I'll hit return. So it's RAST export genome, feature type, in this case now we're looking for RNA, all lowercase, feature DNA, and E. coli anno.gto is our input. And if you hit return, this will give us, we didn't ask it to be put into a file, but this will give us the FASTA formatted file of all of our RNA genes, because that's the feature type we asked for. If we wanted this in GenBank format, we could simply delete feature DNA and add GenBank and hit return. And now we have all of our RNAs in GenBank file. And all you have to do to get those to a file, remember, is to create a file name from standard output. So we'll call this E. coli.rna.gbk. And now we have a GenBank file over here for our RNAs. So in order to use the RAST toolkit default pipeline, there were 3 scripts. There was RAST create genome. This turned our contigs file and FASTA format to the genome typed object format that RAST understands. There was RAST process genome. This took our genome typed object as input and added the annotation data to it as output. So it read in a genome typed object and read out a genome typed object. The third and final command was RAST export genome. And this one read in our annotated genome typed object, and it, depending on which file format we asked for, returned it in the file format that we declared. So then after you've run those 3 in Iris, all that's left is to just download. If you're working in the command line, you're done. Now, after this video, I recommend going to the 3rd video where I'll demonstrate some more advanced use cases for the RAST toolkit.